Greetings, grade 12s. So what we're going to be doing in this video is I'm going to be going through the structure of nucleic acids in general. So what this is going to do is it's going to basically create the understanding, the underpinning, your whole basis of knowledge for the section of DNA, for the section of nucleic acids. So as a reminder, remember nucleic acids is a general group of molecules. There are various forms of nucleic acid that exist, but they all have the same basic structure, which is what we are going to start going into today. So for example, if you look at something like DNA, DNA itself is one type of nucleic acid. There are other forms that exist that we need to know about, but again, they all have the same basic structure. So this is going to create our underpinning for the section of DNA, protein synthesis, and then on towards genetics and genetic engineering. So this is very important that you start at this basis for your understanding. So as a little bit of a harp back towards grade 10 for you guys, I want to discuss this concept of monomers again. So what did we mean by the term monomers? Monomers are the building blocks of compounds. So remember, if we were to break down a much larger compound of something, say we look at the digestive system, for example, you have to break down that burger that you eat, whatever it is, or whatever you're eating for your food, into its basic monomers before it can be absorbed into your body. So essentially, it's the most basic building blocks to create whatever it is. The way I like to think about it is think of Lego. Lego is something that you have to put different Lego pieces together to create Bobby's dream house or your Lego Ninjago, whatever it is that you're wanting to build. But once I've taken all of it apart, I am left with these pieces of Lego, the basic shape of the Lego. And that's really what the monomer is. Now, you can technically argue that you can break down a monomer, a biological compound, even further into its atomic structure. And yes, we do as humans, we take things like calcium and magnesium and phosphate and all those actual elements, and we do use them. But for the purposes of this video, we are going to stop at the concept of a monomer, just so you get the understanding. So as a basic recap, remember the monomer for carbohydrates was the monosaccharides. So that was glucose, fructose, and galactose. When we combine those together, we would create the more complex carbohydrates. For lipids and fats, it was glycerol and fatty acids were the monomers. And for proteins, it was amino acids. Now, proteins are still very important to us. So proteins play a huge role in terms of DNA, and you need to remember that amino acids are the monomers of the protein. So don't forget about the proteins and the amino acids. But for this video, we are wanting then to focus on nucleic acids. And what is the monomer for a nucleic acid? That is a nucleotide. So let's break it down a bit. So a nucleic acid is made up of three components. All nucleic acids have these three basic components. The first is a sugar. Now, pay attention to how I am showing these images to you for when we discuss the structures. Notice with the sugar here, I have used a 5-sided pentagon. And there's a reason for that. Because the sugars that you find in nucleic acids are actually pentose sugars. Each of these would represent a carbon atom. So it's 5 carbon atoms in a ring. If you were to compare this to, example, your table sugar, so like a glucose sugar, is 6 carbon rings around there. So the sugars that we use in nucleic acids are pentose sugars. Now, depending on the different type of nucleic acid, there will potentially be a different type of sugar that is used, but they will all be pentose sugars. But all nucleic acids have a sugar. Then they have a phosphate as the element phosphate, and we like to represent that just by a little circle. And then the third part is a nitrogenous base. So you do not have to worry about the actual atomic structure and the chemical composition of the nitrogenous bases. That you just need to know that it is a nitrogenous base, as in it needs nitrogen as part of it to create so that's why we need that there. In this particular image, I have used a 6-sided figure, a hexagon. You do not if you are drawing this by yourself or you see a picture, it may be something different. This is now just an arbitrary figure that I am using. However, the sugars will generally always be represented as a 5-sided sugar and the phosphate as a little circle. Okay. And again, as well, with the nitrogenous bases like the sugar, they will vary in terms of what the base is. But all nucleic acids will have these 3 basic components. So, we put these 3 together to form the monomer of nucleic acids, which is a nucleotide. So if I was to break down a nucleic acid into its most basic component, that would be a nucleotide. So what does that look like? Well, a single nucleotide is a sugar and a phosphate that are connected to each other, and a nitrogenous base. So this represents a single nucleotide. Again, this 5-sided figure will generally be the sugar. Always use a 5-sided. This will be a circle being the phosphate, and the nitrogenous base can change. You can make it whatever you want based from there. Now, you need to be able to draw this as a sketch. So I could say to you, sketch the structure of a nucleotide. I could ask you to identify different components if I give you a picture. So I could ask you for a whole nucleotide or parts of a nucleotide. You need to be able to do that. So please bear that in mind as part of nucleotide structure. So if a nucleotide is the monomer, we then know that we would link a lot of these molecules together to then form a more complex compound, such as DNA, for example. And what we get when we link many nucleotides together is a polynucleotide chain. So in this sense, poly means many, so many nucleotides linked together. And this is generally what it will look like. So there's 1, 2, 3, 4 nucleotides put together. Now, very importantly, in this single chain of nucleotides, notice where the connection is happening between the nucleotides. It is happening between the phosphates and the sugars. Do you notice this connection here? And the sugars connecting to the phosphate all the way down here. There is no connection between nucleotides of the nitrogenous bases. So you see, there's no connection here. There is no connection between nucleotides, between the sugar and the nitrogenous base. So you see, there is no connection here. The only connection within the same polynucleotide chain is between the sugar and the phosphate. So make sure that you recognize that. So this is a polynucleotide chain. Now remember, the sugar and the phosphate are going to stay the same. So if, for example, I'm talking DNA, it's the same sugar molecule that I'm going to keep having because it doesn't change. It's always the same type of sugar, and the phosphates are going to stay the same. The thing that's going to be different then is these nitrogenous bases. We get different versions of them. So there are 4 different overall nucleotides that you can have in DNA based on which nitrogenous base you have. So these are the 4. So they are thymine, adenine, cytosine, and guanine. So those are the 4 nitrogenous bases. Again, in this instance, whatever this nucleotide is, again, the sugars are staying the same, the phosphates are staying the same, it's just the nitrogenous bases that are different. Thymine, adenine, cytosine, and guanine. Those are your 4. You need to know those, you need to know the words, you need to know how to spell them. So, we can further divide these into either single-ringed or double-ringed bases. So, again, you do not have to worry about the physical structure. You do not have to worry about what physically goes in there, atomic-wise. It's just a single ring and 2 rings. So, the 2 single-ring bases are cytosine and thymine. And the double-ringed bases are adenine and guanine. So you can see there the 2 rings and the 2 rings. How you are going to remember this is completely up to you. If you can create a very clever mnemonic to try and remember, that it would really be good for you from your side. If you can rote learn, even better for you from there. But really try and think of some rhyme or a way that you can try and remember, maybe like D-O-G, double, but the O is kind of like a dog, dag, I don't know. However you want to, maybe dog, dag. If you come up with a really clever way, put it in the comment section below so I'm always looking for new ways to try and remember these different things. But as an example, let's now look at this polynucleotide with the 4 different bases. So here we've got our cytosine, adenine, thymine, and guanine. And then we've got our sugar and our phosphate that are connected, but no connection here and no connection here. So this is one polynucleotide chain. One polynucleotide chain. But when we look at something like DNA, deoxyribonucleic acid, we know that there are 2 chains of polynucleotides that occur. It is a double helix. The double meaning that there's 2 chains of polynucleotides. So what you are seeing here, remember, the sugar and the phosphate stay the same, and we refer to it as the sugar-phosphate backbone. So the sugar-phosphate backbone is what stays the same. What then becomes different is what's happening in the middle with the nitrogenous bases. So how do we put them together? Now, a logical person would probably sit and think, okay, well, if it's containing the code, if I've got one on this side, it would be the same on the other side, because they need to be exactly the same. My DNA needs to be exactly the same. But it doesn't actually work like that. So, when you have your bases combining in the middle, a 2-ringed base will always combine with a single-ringed base. So if on one side of the polynucleotide is a double-ringed nitrogenous base, the same side directly opposite to it would have to be a single-ringed base. So how do these actually work? So, adenine will always join with thymine, and guanine will always join with cytosine. And it works the same the other way. If I've got thymine on the one side, it will be adenine on the other side. If it's cytosine on one side, it will be guanine on the other side. So what do we call this? We refer to this as complementary base pairing. So it's not direct copies, it's not exactly the same. We complement it with the one that fits with it. So what fits with this double? This single. What fits with this double? This single. So please be very aware of that phrase. Complementary pairing is going to be something you really need to be comfortable with. So, and know that A goes with T, G with C. One more thing to add here. These bonds that exist in the middle. So remember, I'll have one polynucleotide chain going down this side and one polynucleotide chain going down this side. I've got the sugars and the phosphates that are connected as the backbone, but in the middle, holding the 2 chains together in the middle, these bonds are referred to as hydrogen bonds. So sometimes you'll see in textbooks that they get referred to as weak hydrogen bonds. And I know the physics people sometimes get a little bit, oh, there's no such thing as weak hydrogen bonds. What are you talking about? But, you know, it's fine. If you refer to it as weak hydrogen bonds, or if you refer to it as hydrogen bonds, you'll be fine. Just know that holding between the nitrogenous bases of opposite polynucleotide chains is hydrogen bonds. So just to finish off, imagine this was our original polynucleotide that I showed you. This is 1. But now if I'm going to have DNA, I'm going to have 2. We're going to do this in complementary fashion. So starting from the bottom, if I have cytosine on one side, what am I going to have on the other side? I'm going to have guanine. And then they will link with hydrogen bonds. If I have adenine, the A, I will then have thymine. And I get the bond. Notice again, I've got the connection happening, sugar-phosphate backbone happening at the same time. If I have thymine on one side, I will have adenine on the other side. Join and join. And if I have guanine on one side, it will be cytosine on the other side. So this, again, was just DNA that I was looking at. If I look at something like RNA or tRNA, all those different things, remember, I will always have a sugar of varying forms, I will always have a phosphate, and I will always have a nitrogenous base that varies. And that is the basic structure of nucleic acids. So please make sure you go through this, have an understanding. Again, this is the base knowledge. If you don't understand this, you're not going to be able to move forward with the other sections that we are doing. Pause, rewind, watch again, make sure you have some notes. Hope you enjoyed that, guys. Keep the studying nice and hard. Let's make sure that we get a solid understanding of this. If you need any help, hit me on the Skype, get me on Teams. We can have a meeting. Otherwise, thanks for watching the video, guys.